Hi, welcome to another screencast. The previous one was on mitosis, which is the basis for reproducing cells, replacing cells, and asexual reproduction. Now we are talking about meiosis, which is the basis for sexual reproduction. And what you want to keep in mind is that the big benefit of sexual reproduction is producing new or unique combinations of genes. So it's all about genetic variability in your offspring. This is what your genes look like. It is called a karyotype. You don't see them very much anymore because we can sequence DNA and it tells you a lot more than just taking a picture of your chromosomes. But this is one of the things people used to do. And you can see that you have chromosomes that look alike. So there are 22 matching pairs of chromosomes. That is because when you reproduce sexually, you get one set of chromosomes from dad and another set of chromosomes from mom. So you end up with 22 pairs of what we call homologous chromosomes. So homologous chromosomes are chromosomes that contain the same genes. So whatever genes are on this chromosome from mom, different versions of all the same genes are on the other chromosome from dad. So if dad has blue eyes, dad's blue-eyed gene is right here. If mom has brown eyes, mom's brown-eyed gene is right there. And the same goes the whole way down the line. Whatever genes are coded for on dad's chromosome number 1, the same genes are coded for on mom's chromosome number 1. So in all of your somatic cells, or body cells, as we just said, you have 46 chromosomes. 22 of them are matching pairs. Right? That's all these. Those are called autosomes. And then in humans, we have 2 sex chromosomes, an X chromosome and a Y chromosome. If you recall, XX is female, XY is male. As you can see, there aren't a lot of genes on the Y chromosome, which makes sense because females don't get one, so you can't have genes that only half the population gets if those genes are important. So then, putting this all together, sort of, because you have 2 copies of each chromosome, we refer to the number of chromosomes in the adult or somatic cell as being the diploid number. So for us, it's 46, 2 copies of 23 chromosomes. If you are going to reproduce sexually, you're going to have a sperm that fertilizes an egg, and all of the genetic material in the sperm will combine with all of the genetic material in the egg. If the egg and sperm had the full complement of chromosomes in the adults, our chromosome number would double every generation, which would quickly become untenable. So we have a process called meiosis that is designed, or that functions, I should say, to cut in half the number of chromosomes so that when sperm and egg get together, you reconstitute the 2n number of chromosomes. So the whole goal of meiosis is to cut in half the number of chromosomes so that egg and sperm can get together and reconstitute the 2n number of chromosomes. This is all text which you can read on your own. So essentially what meiosis is, if you want to break it down, is one round of DNA replication. So you take a regular old cell, it replicates all of its DNA just like before mitosis. Then you go through 2 rounds of cell division, which ends up producing 4 daughter cells with half the number of chromosomes. So if you see up here, you started with 2 chromosomes, you duplicate them, so you basically make enough DNA for 2 cells, but then you make 4 cells, each cell winds up with half the amount of DNA, or half the number of chromosomes in the original cell. We are going to keep meiosis really simple and just talk about what happens during meiosis 1 and what happens during meiosis 2. So to go into a little bit more detail, This is interphase. Before meiosis, just like interphase before mitosis, this cell has two chromosomes. So we've made things very, very simple. Those two chromosomes go through replication, so you end up with two chromosomes that have been replicated, so you have sister chromatids. So you start out with two copies of all of your 2N chromosomes. Then, during meiosis 1, or the first division, the homologous chromosomes all pair up. So if this were a human cell, you would see 23 pairs of homologous chromosomes all lined up at the metaphase plate, and then the homologs are divided between the two daughter cells. So if this is mom's chromosome goes here. If this is dad's chromosome goes there. So at this point now, each cell has two copies of one of the original two chromosomes. Then, in meiosis 2, the sister chromatids separate, so you have two chromatids on this chromosome which separate to give these two daughter cells, and two chromatids on this chromosome which separate to give those two daughter cells. So the copies split, and you end up with each cell having one copy of the N number of chromosomes. And that's the most detail we are going to go into. So you start with one copy of 2N chromosomes, and then you make two copies of your 2N chromosomes. Those chromosomes, or homologs, divide in the first meiotic division. So after meiosis 1, you have two copies of 1N chromosome, and then the chromatids separate, and you end up with four cells that have one copy of the N number of chromosomes. And that is meiosis, as short and sweet as I can make it.